So the cell is the basic building block of all life tissue, whether that be plant or animal. The cells collect together to form tissues, organs, structures, all that kind of stuff. And the basic structure of the cell, the human cell, is roughly the same regardless of the type of tissue. The contents will vary and the function will vary, but the basic structure is the same, and that's what we're going to be looking at in this session. As we go on throughout the semester, we'll look at how the cells vary in terms of their contents and structure, but at first we just want to get the basics down. So you should recognise this diagram, and this is a diagram that featured in the reading that you've done, and it is collecting everything together, looking at all the possible contents of organelles that can be within the cell. But we're going to start by looking at the cell membrane. So again, this is a diagram from your reading, and we're just going to look at the cell membrane in a bit more detail. It's very important. The cell membrane holds it all together. Without the membrane, the contents would just be floating around in amongst all the body tissues. We need the membrane to hold everything together to give it a bit of structure. It also controls passage of substances in and out of the cell in various different ways, which we'll have a wee chat about later. So the cell membrane is sometimes referred to as the plasma membrane, as you can see on this diagram here, or the phospholipid membrane. It's also referred to as a fluid mosaic. It's made up of 4 main components. The first thing to think about is the phospholipids. So the phospholipids, as you can see from the pointed here, are the main sort of building blocks of the plasma membrane. They come in a sort of head on a stick type form, and this is what this little key is telling you here. And they're arranged in a double layer. And one of the reasons why I refer to the plasma membrane as a fluid mosaic is because these phospholipids can all move together in a sort of wave-like fashion so that the cell can change shape. It can stretch, it can shrink back down, it can squeeze through tight spaces, all that kind of stuff. So the head and the tail of the phospholipids are arranged in a very specific way. So we'll just highlight a couple of things here. You can see that they're sort of back to back. So we have all the heads either on the outside of the cell membrane in contact with the interstitial tissues, or on the inside of the cell membrane pointing in towards the cell. And then the tails all link together in the middle. So if we go with a different colour, we can see the tails are all the way along here. And they come together and form this sort of tight mosaic. We use the terms hydrophilic and hydrophobic to refer to the heads and the tails. You can see that we talk about the heads, which are on the outside, as hydrophobic, and the tails in the central core of the fluid mosaic as being hydrophobic. So I want you to go and spend some time researching those terms. A straightforward Google or dictionary search will find them. And when you find out what those terms mean, have a think about why we want those to be that way. Why do the heads need to be hydrophilic, and why do the tails need to be hydrophobic? And what kind of function does that serve when it comes to the main roles served by the plasma membrane? So the phospholipids make up the fluid structure of the cell membrane, but we also need some other things in here to give it structure and to serve other functions. So another large component of the cell membrane is the proteins, which I'm just highlighting here with these blue marks. So these are big solid chunks that you find in amongst the phospholipids. They can be all the way through, so like this big one at the front here, and we refer to them as integral. So they're from the outside right through to the inside of the cell. They can also be what we call peripheral, which is what we would call these two here. They are on the outside or the inside, but they're not going all the way through. Peripheral proteins can also be just found just within the tails of the phospholipids and not coming out through the heads at all. Now, the proteins serve lots and lots of functions. Part of it is to do with just providing a bit more structure. We've said that the phospholipids are fluid, and that's an advantage to some extent, but there may be times when you want a slightly more rigid cell. So the more protein molecules that you have within the cell membrane, the more rigid that membrane will be. They're also concerned a lot with passes of substance in and out of the cell. This cell membrane provides a barrier to keep things in or out of the cell, but there are things that need to come in and there are things that need to leave. So a cell needs its raw materials, its nutrition to come into the cell in order to allow it to perform its functions. And then it also has waste products and also anything that it's manufactured that need to leave the cell so that they can either be disposed of or taken to where they need to be. So it's really important that we can have mechanisms by which sometimes quite large molecules can enter or leave the cell. And the proteins are concerned very much so with that. And when we move on to talk about the task that you've been set for this session, that will come into play a little bit more. Other structures that these proteins serve is they can be enzymes. So enzymes that help to catalyse processes that are going on within the cell. If the protein is peripheral and has on the internal aspect of that cell membrane, then the cell can use that as an enzyme to catalyse its processes. When they're on the external of the cell membrane, then they're often binding sites and receptors for other components within the tissues to link onto that cell, either to help it perform its processes or for protection and all these kind of things. So it just allows other things to hang on. If you think about the phospholipid as being a really smooth, slippery surface, then it would be very hard for anything to grab onto. And these peripheral proteins on the external can be like a wee handhold to allow other things to link up with that particular cell. So they serve lots of functions, more complex than we need to know, but it is important that you understand that the proteins are a really important part of the cell membrane. The next thing I want to think about is the carbohydrate chains, and we can see those up in this area here. If we highlight those so that that's a little bit more obvious. These little chains that are hanging off the proteins. So carbohydrates help the cell give its identity. All our cells have a flag, a name tag, shall we say, that allows our body to say, oh yeah, that's mine, that's happy to be there. And it means that when something enters the body that we don't want to be there, whether that be a virus or a bacteria or even just an old cell of our own that we're trying to get rid of, they can look at that flag or that name tag and go, oh no, that's not for me, and go through a process to do something about it. Also, even our own cells itself have a name tag that says that this is what I do and where I should be. And that's one of the main functions that the carbohydrate chains within the cell membrane serve. They come in two forms. You can have carbohydrate chains attached to the protein molecules, and that's what you can see in this diagram here. And we refer to those as glycoproteins. So the glyco refers to the carbohydrate, and the protein refers obviously to the protein that the carbohydrate is attached to. You also find carbohydrate chains attached to the heads of the phospholipids on the external of the cell membrane, and we call those glycolipids. So glycoproteins attached to the protein molecules and glycolipids attached directly to the heads of the phospholipids. But either way, they serve the same function, and that's to allow the cell to be recognised. And then the last thing that we find in the cell membrane, it's a little bit harder to see, is cholesterol, sorry. Now, we think of cholesterol as a bad thing. We think, oh, my cholesterol is a bit high, or I'm eating too much highly processed food, red meat type thing, and we need to bring the cholesterol down. And that's absolutely certain true to a certain extent. However, we do need a small amount of cholesterol to serve a really important function in our body, and it's about holding stuff together and giving us structure. So we have cholesterol chains, short cholesterol chains, embedded within the tails of the phospholipids, and they just help it all stick together. A little bit like glue or twine that's wrapped around things to hold it together. Without these cholesterol chains, when the cell needed to change shape, so stretch or come back to normal or to squeeze through a tight space, the phospholipids would be at risk of coming apart, and then obviously the cell contents would be spilled out and we would lose that cell. So whilst we are a little bit scared of cholesterol in our diet, we do need it within our bodies to perform that function. So that's really going through the structure of the plasma membrane. Just to recap, we've got our phospholipids as a fluid mosaic, a double layer, heads on the external of the membrane and the tails pointing in towards the middle. And I've asked you to go and have a wee read and see what you can find out about the fact that the heads are hydrophilic and the tails are hydrophobic and the relevance of that. We've then got our protein molecules, which can either be integral or peripheral, and they allow the cell to have the membrane to have a little bit more structure, to become a little bit more rigid. They also allow controlled passage of substances or larger molecules in and out of the cell, and they can act as enzymes and binding agents. We have our carbohydrate chains to give identity to the cell. And then finally, we have our cholesterol chains within the tails of the phospholipids to hold it all together.